All right, good afternoon, everyone. Hope you're all doing well once again. I just wanted to do a walkthrough of this lab because unfortunately it's very difficult to do on your own because there are a lot of difficult equations to deal with. For some, it may be easier, but if it's not that easy for you, that's totally fine, and I would recommend watching this video. So in your lab manual, we are in chapter 10 about spatial relationships. Sorry, I cannot make this go away. We're going to talk about organizing data and biotic factors, limitations on populations, and things like that. We already discussed these factors in the PowerPoint, so this is introductory information. What we want to focus mainly on here is that we have 3 different types of dispersion, and these could be caused by many different factors, whether they're abiotic or biotic, that being random, even, or clumped. And we're going to use 3 different tests in the simulation in order to see each type, hopefully, of dispersion. The first of which is called the Clark-Evans nearest neighbor method. This is what may be confusing to some because there are data sheets that talk about GPS coordinates, species A, species B. We would have gone outside and actually collected distances between nearest neighbors of species, but in this case we are not. We are just using the simulation, and those data sheets are on a different page. They're tables 10-1 and 10-2, and we'll see them later in this video. This explains everything the Clark-Evans nearest neighbor method test uses, and honestly, I have not used this test or any of the following tests before this lab, if that tells you how prominent they are in biology. Okay, so we're testing to find out if R is significantly different from 1 by using a t-test. So here, we're not going to do this part of 175, don't worry. So start at 6 by putting down your hypotheses. So they have them here in English. In math, it would simply be the null would be that R basically equals 0. So 1, R equals 1. And the alternatives could be R is greater than or equal to 1. Note that significantly is a key word here. There are a lot of different notations here that are on the last lab page that you need to know what they mean. A lot of calculations you have to do. The density of the plot is something that I had not provided for you, but I do have that now, and that's provided in the Excel. So this is finally how you come up with that final R, but let's move on to the other test. So after you find R, you want to see if that value is statistically significant using that t-table. The second test is called the variance mean ratio method. So ignore this, go down to here. These are your hypotheses. The variance mean ratio not significantly different from 1. So this notation equals 1, or this notation here is greater than or equal to 1. Here are some of the equations that are used, variance over mean. The data for both this test and the following are both collected in table 10-2 and from doing the simulation, okay? So once again, you're going to use the t-test to determine significance here. The interspecific association test, 178, is the final test we'll be doing. And this test to see if there is a positive or negative association between the species. In this case, we essentially have 2 quote-unquote species because in the simulation there are 2 differently colored dots that you'll compare between. Don't worry about that. We're not going outdoors. Here are those hypotheses once again. You can just do like negatively is minus, A and B equals minus, A and B equals positive. I'm not too worried about the mathematical statement as well for that one. The data set is a bit different for this one, so you're going to have to construct this yourself. So you're going to have to, you can use Excel to do this. Notice it's kind of like a chi-square, but these columns, or these cells are different, okay? Pay close attention to that. And instead of the class data set, you all are collecting your own data. Okay, so let's keep going. The first thing you're going to turn in out of 3 separate assignments is the pre-lab. It's pretty easy. You just want to find 3 different pictures showing these different distributions. You can find those online. And provide a hypothesis of potentially why they are this way. Even, so even could be people waiting in line to get groceries 6 feet apart. Random, and which is the most common in nature and why? What do you think is the most common in nature? Okay, so don't worry about these data sheets. You're not going to fill these in. Nope, a lot of red X's. But here's where we get to the online simulation part, where you have to go to BioViewer. This is the second of the things you're going to have to turn in. So there are full-length instructions as well as another video on how to access these assignments. So this is 185. It starts with instructions and continues on with instructions until you begin with table 10-1. So you can do 25 throws, I believe. Sorry about that. Anyway, that's something else. These are the distances. So you're measuring distances between the neighbors here, the dots, and recording them by pressing add point and going to the next one. And that's where you'll put the distance there. So let's look at that real quick on BioViewer. And what we see here, when I just type in BioViewer into Google, my first option shows me this. Usually this doesn't work due to Flash, so let's click on BioViewer. I'm going to open now since I've already used it. Remember here that the password is anatomy. And I'm going to go population ecology, nearest neighbor, quadrant, plus sign. Just doing this very quickly. There's a more detailed explanation in another video. Add point, add point. So distance 1 will be from here to its nearest neighbor. My cat, sorry, to here, which is 242. Add point here to here. It's not really its nearest neighbor, but hold on one second. I think my cat is okay with me talking again, sorry. Let's exit from this. Do a new screen share once again. Back to, so every distance you get, so 242, whatever distance will go here. You record it until you get 50. And then for the second test, this is your data sheet here, where you notice both the presence and absence of species as well as the number, which are basically the same thing. All right, now I'm petting my cat with my foot. So for the second test, you can click on either of these four. Click on this one and go to quadrants here, zoom in, and I now know the size of these plots. So species A and species B are these two differently colored dots. So let's go back once again and do a new, oh, my screen share was paused. Don't honestly know where that stopped. Okay, I think this is where we're at. Sorry. Please ask me questions if you're worried about this. Let's go down. 188 empty. Number 3, this is the final thing you're going to have to turn in. This is interpreting all of your data into these lab notes. It's only one page. So you already have the hypotheses and what dispersion is supposed to be based on that PowerPoint. Biological conclusion just means, is it random, even, or clumped? And you will have to do this chart similar to what was in the lab manual, and it's just a simple chi-square chart. And instead, you will use the chi-square table for this comparison. Okay. I may need to make another video for clarification again, but hopefully this helps a little bit. So you have 3 assignments to turn in, and I'm going to upload an Excel that will help you with the calculations. So please email me still if you need help with this, and hope you all are doing great. Have a wonderful rest of your week. Thanks.